Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello, and welcome to the June 2020 summary of mostly cerebrospinal fluid, or CSF, biomarkers in the context of Alzheimer's disease. My name is Elyn Rowe, and I'll be your host for this episode. I say mostly CSF biomarkers because some of these papers that I'll be covering also focus on neuroimaging data or have a plasma biomarker or two thrown in, but we're mostly talking CSF today. As you may have heard me say already in the June 2020 Blood Biomarkers episode, I acknowledge that I'm a little late coming out with these, as it's now October. So sorry for the delay. Things with grad school have been heating up lately, and I've been pretty swamped trying to get the ball rolling again after the COVID lockdown. But I'm slowly getting back into a routine, and I'm definitely going to try to be better with staying on top of creating this content for you guys. So a couple of notes before I jump into the hefty feat of summarizing 24 new papers for you. In this episode, I sometimes refer to the classic panel of AD biomarkers, and these are the usual suspects of A-beta 40 or 42, total tau, phospho-tau, which I often call p-tau, and neurofilament light, which is also known as NFL, and that's a marker of axonal damage. These markers are widely known as and accepted as being useful in the early detection and diagnosis of AD, but there's lots of ongoing work to validate their utility in different cohorts and contexts, as you'll see in this episode. I also want to mention that I sometimes jump right to reporting the discriminative capacity of a biomarker or model by saying what the AUC value is. If you aren't already familiar with biomarker work, AUC stands for area under the curve, and it's a summary number used to describe the receiver operating characteristic, or ROC curve. These curves demonstrate how well a biomarker or model can discriminate between two groups, like say patients with AD and healthy controls. And the best AUC value, indicating a perfect discrimination, so perfect sorting by the biomarker or model, that value would be 1. So anything close to 1, like over 0.8 let's say, indicates that the model or biomarker is pretty good. So now that we've got that covered, I'll begin with the dive into these papers, with those focused on identifying or validating novel CSF biomarkers, so not our beloved classics, for AD. So starting off with a broad strokes paper, we have first a paper titled Proteome Profiling in Cerebrospinal Fluid Reveals Novel Biomarkers of Alzheimer's Disease. This is by first author Bader and last author Mann, published in Molecular Systems Biology. This study aimed to identify new CSF biomarkers that could distinguish patients with AD. They used banked CSF from 3 independent cohorts of patients with AD and controls, and with a mass spectrometry workflow, they were able to quantify over 1,000 proteins from only 40 microliters of CSF per patient. Their study found that the proteins that were most different between AD patients and controls were known biomarkers, such as tau, SOD1, and PARC7, proving their method was effective. But they also identified a host of other biomarkers consistent across the 3 independent cohorts of patients, which are detailed in their paper. Interestingly, they found a consistent protein signature indicating glycolysis in AD patients. The paper has a really nice graphical abstract and figures and tons of data to comb through, so definitely be sure to check it out. Next is a paper that looked at a bunch of different sterols as candidate biomarkers, and it's called Cerebrospinal Fluid 7 Keto Cholesterol Level is Associated with Amyloid Beta 42 and White Matter Microstructure in Cognitively Healthy Adults. So it's pretty well known in the field now that abnormal cholesterol metabolism is a big player in AD, and it can actually change the composition of the neuronal membrane to promote A-beta production. So since cholesterol turnover is important for maintaining axons and white matter microstructure, this group aimed to demonstrate a relationship between oxysterols like cholesterol, white matter microstructure, and other AD biomarkers. They wanted to prove that this relationship happened early, before symptom onset, so they used 153 healthy adults in their study. In each participant's CSF, they measured a series of oxysterols and the classic AD biomarkers, and they used diffusion tensor imaging to visualize white matter microstructure. They found that higher levels of 7-ketocholesterol was related to lower A-beta 42, indicating less clearance and greater AD pathology. Interestingly, they also found that levels of 7-ketocholesterol moderated relationships between features of white matter microstructure seen on DTI and other AD biomarkers, including the axonal marker neurofilament light, or NFL. These data ultimately support the involvement of 7-ketocholesterol in maintaining axonal integrity and provide more evidence for the involvement of cholesterol metabolism in the generation of A-beta. Now we have a couple of papers that evaluated synaptic markers, so starting with a paper called Dissection of Synaptic Pathways Through the CSF Biomarkers for Predicting Alzheimer's Disease. This one's published in Neurology by first author Tibble and last author Bleno. This study looked at synaptic biomarkers in the CSF and evaluated their potential to discriminate between AD and non-AD disorders. It was a retrospective cross-sectional study in which 243 participants who had CSF assessments for neurocognitive decline were included, and that ended up being 81 patients with AD, 30 with MCI due to AD, 49 with MCI due to other causes, 49 with other dementias, and 35 controls. Synaptic biomarkers and the classic AD biomarkers were measured in CSF using ELISA or mass spectrometry. They found that all synaptic biomarkers were significantly increased in patients with AD or MCI due to AD compared to other groups, and they could all discriminate AD dementias from other dementias with an AUC over 0.8. The synaptic marker SNAP25AA50 had the highest discriminative power with an AUC of 0.93 for discriminating between AD dementia and other dementias or controls, and an AUC of 0.9 for MCI due to AD versus controls. They also noted that higher levels of the synaptic markers were associated with ApoE4 homozygotes. This definitely puts synaptic markers on the map, and it will be interesting to see whether these findings can be validated in other larger cohorts. So sticking with synaptic markers to some extent, we have another paper called Cerebrospinal Fluid YKL40 and Neurogranin in Familial Alzheimer's Disease, a pilot study. This is by first author Thordar Dottir and last author Graf, published in the Journal of Alzheimer's Disease. So this pilot study investigated levels of YKL40 and neurogranin in the context of familial or early onset AD. As some context, because I wasn't super familiar with these markers, YKL40 is a glycoprotein that's been found to be a marker of neuroinflammation, and neurogranin is a synaptic protein. The authors measured these analytes in the CSF of 14 individuals carrying a familial AD mutation and 17 non-carriers from the same families. In this pilot study, they didn't find any differences in the markers when they compared the pre-symptomatic mutation carriers with the controls, but YKL40 did positively correlate with the expected years to symptom onset and age in both groups, while neurogranin didn't really show any correlations. They also had a subgroup that had more than one CSF sampling over a period of time, enabling the authors to look at some longitudinal trends. With this, they found an increase in YKL40 in the mutation carriers as they got closer to symptom onset, but again, no relationships with neurogranin. With these data, it seems like YKL40 may be a good marker for predicting symptom onset in early onset AD as well. So next, onto a hormone as a potential CSF biomarker. We have a paper called Cerebrospinal Fluid Iresin Correlates with Amyloid Beta, Bdnf, and Cognition in Alzheimer's Disease. This is by first author Lorenco and last author Ferreira, published in Alzheimer's and Dementia. So Iresin is a hormone that is classically known for its role as a regulator of metabolism in the periphery, but it's recently been shown to protect synapses and improve memory in mouse models of AD. This group set out to figure out whether levels of Iresin are related to classic AD biomarkers and cognition in a clinical context. Their cohort consisted of 14 patients with AD and 25 cognitively healthy controls, in which they measured Iresin, brain-derived neurotrophic factor, or Bdnf, Abeta42, and total tau in CSF. They also performed the MMSE to evaluate cognitive function. They found that Iresin had a significant positive correlation with Bdnf, Abeta42, and cognitive scores, but not with total tau. It'll be interesting to see whether levels correlate with phosphorylated tau or synaptic markers in larger cohorts. Next up is a paper that leveraged the APP-PS1 mouse model to gain insight into biomarkers for early AD. And it was called Hippocampus Proteomics and Brain Lipidomics Reveal Network Dysfunction and Lipid Molecular Abnormalities in APP-PS1 Mouse Model of Alzheimer's Disease. This is by first author Zhang and last author Tan, published in the Journal of Proteome Research. This paper is a multi-omics-based study in the APP-PS1 mouse model that aimed to identify new protein or lipid biomarkers in early-stage AD. The The group sacrificed APP-PS1 and wild-type mice at 7 months of age and used their full brain homogenate for lipid extraction and their dissected hippocampus for protein extraction. The extracts were then analyzed with mass spectrometry, and through this the group identified 231 proteins and 11 lipids that were significantly altered in the APP-PS1 mouse brains. Specific pathways that were affected included complement and coagulation cascades, glycerophospholipid metabolism, and PI3K-AKT signaling. While there are limitations to this study given that it was in a mouse model of early onset AD, the work ultimately sheds light on pathways that may be affected in early stages of AD, which is key in the search for novel biomarkers. So keeping with the trend of translational animal models, next we have a paper called Age-Associated Changes in Amyloid Beta and Formaldehyde Concentrations in Cerebrospinal Fluid of Rhesus Monkeys. This is by first author Lee and last author Hu, published in Zoological Research. So since rhesus monkeys are so close to humans on the evolutionary tree of life, they're pretty good models of neurodegenerative disease as well. It's already been demonstrated that they show similar hallmarks of aging and to some extent neurodegeneration, but their CSF profile hasn't fully been characterized. This study set out to determine how formaldehyde levels in rhesus monkey CSF change with age and whether these changes are correlated with A-beta concentrations. The authors performed a cross-sectional study on 56 monkeys aged 4 to 26 and measured levels of A-beta 40, A-beta 42, and formaldehyde in their CSF. They found that there were significant decreases of both forms of A-beta and a significant increase of formaldehyde with age. They also found that the increase in formaldehyde was negatively correlated with A-beta levels in aged monkeys, but not in young or middle-aged monkeys. Ultimately, these findings are similar to what's seen in humans, reinforcing that rhesus monkeys are good models of neurodegenerative disease. Next, we have a paper called Association between Cerebrospinal Fluid Biomarkers and Age-Related Brain Changes in Patients with Normal Pressure Hydrocephalus. This is by first author Tagdiri and last author Tartaglia, published in Scientific Reports. So this paper isn't directly related to AD, but it does use biomarkers to provide some insight into mechanisms of A-beta clearance. The paper focuses on a condition called normal pressure hydrocephalus, which is a term to describe a surplus of cerebral spinal fluid that builds up in the brain ventricles. This buildup results in an excess of pressure on the brain and can result in clinical symptoms similar to dementia. That said, it's often misdiagnosed as AD. So this study aimed to use classic AD CSF biomarkers, A-beta 42, total tau, and p-tau, to distinguish between normal pressure hydrocephalus, AD, and healthy controls. Their study included a group of 38 patients with probable and 34 patients with unlikely normal pressure hydrocephalus, 30 patients with AD, and 29 cognitively normal controls. They found lower levels of A-beta 42 and p-tau in probable normal pressure hydrocephalus compared to normal controls and compared to AD. Overall, the study supports the hypothesis that an increase in interstitial space, as seen in normal pressure hydrocephalus, results in better A-beta 42 clearance into the CSF. So not your classic AD-focused paper, but definitely some interesting insight. Next up is a paper called Downregulation of Synapse-Associated Protein Expression and Loss of Homeostatic Microglial Control in Cerebrospinal Fluid of Infectious Patients with Delirium and Patients with Alzheimer's Disease. Bit of a mouthful, that one. So first author for that is Peters Van Ton, and last author is Abdo, published in Brain Behavior Immunology. So given the relationships between systemic inflammation in both delirium and dementia, these authors set out to determine whether patients who develop delirium have an overlap with those with AD in terms of their CSF biomarker profile. They measured protein expression signatures using a proximity extension assay in CSF from 15 patients with infectious delirium, 29 patients with AD, 30 infectious patients without delirium, and 15 non-infectious controls who didn't have any neurological disease. They found that 8 inflammatory proteins, including CX3CL1, which is involved with communication between neurons and microglia, were significantly upregulated in both delirium and AD compared to other groups. They also found that 23 proteins were downregulated, including CD200R1, which is another marker of neuron microglia communication, and also a cluster of proteins related to synaptic formation and function. All of these results were partially mediated by age, which is a common risk factor for both delirium and AD. But overall, it seems that synapse pathology and alterations in neuron microglia communication may be common factors between the two disorders. On to a couple papers aiming to distinguish between types of dementia. We first have Electrophysiological Differences in Cortical Excitability in Different Forms of Dementia, a transcranial magnetic stimulation and laboratory biomarker study. This is by first author Kedder and last author Goma, published in Neurophysiologie clinique. As you can probably guess from the title, this study mainly focused on identifying differences in cortical excitability in different types of dementia. But they also measured plasma levels of transforming growth factor beta 1, or TGF beta 1, in their cohort. The cohort they used included 15 patients with each type of dementia, including AD, vascular dementia, or Parkinson's disease dementia, as well as 25 control subjects. The authors examined cognitive status with the MMSE, a series of outputs related to excitability, and serum levels of TGF beta 1. They found significant neuronal hyperexcitability in all groups with dementia compared to controls, and significantly increased levels of TGF beta 1. They also found that there were longer contralateral and ipsilateral silent periods in AD and Parkinson's disease groups compared to the others, and that there was a significant negative correlation between serum TGF beta 1 and ipsilateral silent period duration in AD specifically. Overall, these data suggest that cortical activity may be unique in different forms of dementia, and that TGF beta 1 may be associated with neuronal dysfunction in dementia and perhaps particularly in AD. So sticking with parsing apart different types of dementia, next we have differential levels of neurofilament light protein in cerebrospinal fluid in patients with a wide range of neurodegenerative disorders. This is a paper by first author Delaby and last author Leo, published in Scientific Reports. So this study looked across the board at a bunch of different neurodegenerative diseases to evaluate the utility of NFL as a biomarker in a range of disorders. The authors measured NFL and CSF from 535 patients in the St. Po Initiative on Neurodegeneration, also known as the SPIN cohort, which included patients with a range of neurodegenerative diseases as well as cognitively normal participants. As expected, they found that all neurodegenerative diseases had increased levels of NFL compared to the controls, but the highest levels were observed in patients with ALS, frontotemporal dementia, corticobasal syndrome, and progressive supranuclear palsy. They also found an association between NFL levels and cognitive impairment in patients with AD pathology specifically. This work supports the use of NFL as a marker for neurodegeneration in a wide range of contexts and highlights its relationship with cognitive status in the context of AD. Okay, so up until now it's been almost exclusively CSF biomarkers, but now we're on to a section where things are a little more convoluted, and many papers use multiple different indicators of AD, including some CSF biomarkers and some neuroimaging. A few of these may have slotted better into other episodes, such as our amyloid and tau detection episode, but we're still hammering out some kinks in our sorting methods, so bear with us. First up, I have a few papers characterizing how indicators of AD change over time or between clinical phenotypes. And the first one is titled, The Dynamics of Biomarkers Across the Clinical Spectrum of Alzheimer's Disease. This is by first author Hadiji Krisanthu and last author Anderson, published in Alzheimer's Research and Therapy. As stated in the title here, the group looked at the rate of change and temporal order of AD indicators throughout the AD continuum, from preclinical to severe AD. The group used 27 different markers, including a range of cognitive scores, fluid biomarkers, and neuroimaging, from 2,014 participants in the ADNI cohort to build stochastic models that could estimate the expected time to reach different clinical disease states for people in different risk groups. Their model found that detectable memory dysfunction could occur up to 3 decades before dementia onset, which was closely followed by changes in CSF A-beta levels. They also noted that hippocampal atrophy can be observed as early as the initial detection of A-beta accumulation, and that NFL was the only protein with notable early changes in plasma. Further, brain hypometabolism and changes in the levels of total and p-tau seem to start later, at about 14 years before the onset of dementia. They provide lots more insight into the progression of the disease with their model built from the rich dataset, so definitely check it out. Next are a couple of papers that solely focus on CSF biomarkers, with the first being amyloid beta, tau, synaptic, neurodegeneration, and glial biomarkers in the preclinical stage of the Alzheimer's continuum. This is published in Alzheimer's and Dementia by first author Mila Aloma and last author Molinuevo. So this study delved further into characterizing the CSF biomarker profile of preclinical AD. The authors used the Neuro Toolkit and Alexis immunoassays to measure the classic AD biomarkers, as well as neurogranin, soluble TREM2, YKL40, IL6, S100, GFAP, and alpha-synuclein in cognitively normal patients from the larger Alpha Plus study. They found that total tau, p-tau, and neurogranin increased with age only in A-beta positive individuals, while NFL and the other glial biomarkers increased with aging in general. When they modeled their biomarkers to proxy disease progression, they found that the first change was a decrease in the ratio of A-beta 42 to 40, followed by phosphotau, total tau, neurogranin, and to a lesser extent NFL and the glial biomarkers. So yet another study showing this pattern in progression and reinforcing the classic biomarker targets to identify individuals in early stages of AD. Sticking with CSF biomarkers only, we have a paper called Amyloid Beta Modulates the Association Between Neurofilament Light Chain and Brain Atrophy in Alzheimer's Disease. This is published in Molecular Psychiatry by first author Kang and last author Rosa Neto. Okay, so back to our old friend NFL. These authors wanted to figure out how well neurofilament light can reflect regional neuronal injury in the context of amyloid pathology in AD. It was a hybrid study that used 160 patients with MCI, 73 patients with AD, and 83 cognitively healthy controls from the ADNI cohort, as well as 13 rats, 5 wild type, and 8 that were McGill R-Thi1 APP transgenic. In the clinical portion of their study, they found that there was reduced gray matter density in the hippocampus and posterior cingulate cortex in MCI or AD patients who were A-beta positive, and these reductions were significantly associated with increased NFL in the CSF and plasma. They then validated these results in their transgenic rat model. Ultimately, these data reinforce the use of NFL as a marker of neuronal injury and demonstrate that reduced gray matter density and elevated NFL levels are associated with AD-vulnerable brain regions in the presence of amyloid positivity. Next up is one that looks at different AD behavioral phenotypes, and it's appropriately called Longitudinal Neuroimaging Biomarkers Differ Across Alzheimer's Disease Phenotypes. It was published in Brain by first author Cintini and last author Whitwell. The study aimed to determine whether longitudinal patterns of tau PET scans can inform on the phenotype of the patient, being either the classic amnesic phenotype or atypical phenotypes such as logophenic progressive aphasia and posterior cortical atrophy. To do this, they used a cohort of 18 patients with typical amnesic AD, 17 with posterior cortical atrophy, and 22 with logophenic progressive aphasia that had baseline and 1-year MRI and tau PET data. And they also had a control group of 49 cognitively healthy individuals. The topography of longitudinal tau accumulation and atrophy differed across cognitive phenotypes, with key regions of tau accumulation in the frontal and temporal lobes for all phenotypes, and key regions of atrophy in the occipitotemporal regions for posterior cortical atrophy, left temporal lobe for logophenic progressive aphasia, and medial and lateral temporal lobes for typical AD. They also found that baseline tau uptake in the PET scans mapped better to clinical phenotype than longitudinal tau data or MRI did, indicating the likely causal role of tau as a driver of clinical dysfunction. The next paper is titled CSF Biomarkers in Olmsted County, Evidence of 2 Subclasses and Associations with Demographics. This was published in Neurology by first author Van Harten and last author Peterson. So this study aimed to tease apart relationships between individual CSF biomarkers and their associations with ApoE4 genotype, demographic variables, vascular variables, and clinical diagnosis in people without dementia. The authors used 774 participants from the Mayo Clinic Study of Aging, including 71 with MCI, and the remaining were cognitively unimpaired. They measured A-beta 42, total tau, and p-tau 181 in CSF using immunoassays, and they used linear regression models to investigate the associations with the other variables. Of note, they weighted the results back to the Olmsted County population in Minnesota, as that's what they were specifically interested in. With their models, they distinguished 2 different subgroups in the study population. The first had a strong positive correlation between A-beta 42 and p-tau, while the other had a weaker correlation between the two and was associated with older age ApoE4 genotype and a diagnosis of MCI as well as elevated amyloid PET. Overall trends included that ApoE4 genotype and MCI were associated with A-beta 42 levels, while age was associated with the ratio of p-tau to total tau. They didn't find any associations with sex, education, or vascular risk factors in this study. On to improving and further understanding staging of mild cognitive impairment, we have a paper called Mild Cognitive Impairment Staging Yields Genetic Susceptibility, Biomarker, and Neuroimaging Differences. This is by first author Moore and last author Jefferson, published in Frontiers in Aging Neuroscience. So Alzheimer's disease is pretty well divided into severity stages, but this isn't really the case for MCI, which is often lumped together despite clinical and prognostic differences. This group set out to characterize differences in genetic, CSF, neuroimaging, and neuropsychological markers across MCI stages that were derived from clinician assessment. The study leveraged participants from the Vanderbilt Memory and Aging Project, including 18 classified as mild, 89 classified as moderate, and 18 classified as severe MCI that were all within the same age range. Participants had baseline cognitive assessment, MRI and CSF collected, and cognitive testing and MRI was repeated at 18 months, 3 years, and 5 years. At baseline, the authors found that the 3 stages differed on ApoE4 status, with more ApoE4 carriers in the moderate or severe stages than the mild stage. Also on CSF A-beta levels, with more A-beta 42 in the mild compared to the moderate or severe stages, and with other markers, with more total and phosphorylated tau as well as neurogranin in severe compared to mild or moderate stages. With their longitudinal data, they found that MCI stage related to greater longitudinal cognitive decline, hippocampal atrophy, and inferior lateral ventricle dilation. All of these differences are likely attributable to baseline differences in amyloidosis, tau, and synaptic dysfunction in the different stages, but this paper solidified the importance of MCI staging in describing the underlying pathology. So to cap off this mini segment on longitudinal changes in AD indicators, we have a paper focused on individuals with Down syndrome, titled Clinical and Biomarker Changes of Alzheimer's Disease in Adults with Down Syndrome, a cross-sectional study. And it was published in Lancet by first author Forti and last author Leo. So as a preface, which I gave in the blood biomarkers episode as well if you listen to that, Down syndrome is caused by trisomy 21, and the amyloid precursor protein gene is on chromosome 21. So people with Down syndrome often develop considerable A-beta plaque loads as they age, and many also develop the cognitive dysfunction characteristic of Alzheimer's disease as well. This makes people with Down syndrome uniquely poised to provide insight into AD. So building on this, the authors characterized the order and timing of changes in biomarkers of AD in a population of adults with Down syndrome. The study enrolled 388 participants with Down syndrome, 257 of which were cognitively stable, 48 who had prodromal AD, and 83 who had AD dementia. And they also had 242 controls who did not have Down syndrome. They found that CSF A-beta 42 to 40 and plasma NFL values changed in individuals with Down syndrome as early as in their 30s, and amyloid PET uptake changed in their 40s. Tau PET and CSF p-tau changes occurred later, usually in their 40s, followed by hippocampal atrophy and changes in cognition in their 50s. They also found that symptomatic AD prevalence increased with age in individuals with Down syndrome, reaching 90 to 100% by their 70s. So this was a very informative characterization that draws a lot of parallels with sporadic Alzheimer's disease. Next, I have a couple of novel assays for AD biomarkers, and the first is a paper that describes a new assay that was of particular interest to me since it relates a bit to my own research. And it's titled Simple and Fast Assay for Apolipoprotein E Phenotyping and Glycotyping, Discovering Isoform-Specific Glycosylation in Plasma and Cerebrospinal Fluid. This was by first author Hu and last author Nedelkov, published in Journal of Alzheimer's Disease. Okay, so this was a cool one. The Alzheimer's developed a new mass spectrometry-based immunoassay to determine the impact of ApoE genotype on the relative abundance of ApoE isoforms and their specific glycosylation patterns in CSF and plasma. So really doing the most here, and maybe I'm just so hyped because I love ApoE. So glycosylation is a post-translational modification of adding sugar groups onto a protein, and determining glycosylation patterns on ApoE has become a hot topic for a few reasons. ApoE in CSF is heavily glycosylated, and there may be differences in glycosylation depending on the ApoE isoform, which could in turn affect the function of the protein. So the authors used their assay to measure total glycosylation and isoform-specific glycosylation in paired plasma and CSF from a group of 22 cognitively normal older individuals who were either homozygous for ApoE3 or ApoE4, or heterozygotes of 3,4, 2,3, or 2,4 genotypes. They found that in heterozygous individuals, the ratio of the abundance of different ApoE isoforms differed between plasma and CSF, with the ApoE4 protein being more abundant compared to E3 or E2 in the CSF compared to plasma in ApoE4 carriers. They also found isoform-specific differences in glycosylation in both CSF and plasma, with ApoE2 having the most glycosylation, ApoE3 having less, and ApoE4 having the lowest. There's a lot more to unpack in this paper regarding glycosylation, and I could spend another 10 minutes talking about this paper probably, but we should move on. Overall, their assay is a great new tool for investigating the impact of ApoE isoforms and ApoE glycosylation as fluid biomarkers for AD. So definitely check out this paper if you're subscribed to ApoE like I am. Next is a paper that is also oddly related to me, in that 2 of my supervisory committee members are authors on the paper. The paper is titled An Automated Clinical Mass Spectrometric Method for Identification and Quantification of Variant and Wild-Type Amyloid Beta 1-40 and 1-42 Peptides in CSF. This one's published in Alzheimer's and Dementia by first author DeMarco and last author van der Gutten. So their study was focused on the development of a new mass spectrometry-based assay to quantify wild-type A-beta peptides and also detect variant A-beta peptides in human CSF. The assay was validated with A-beta 42 calibration using certified reference material, and sample preparation was fully automated. The assay was able to successfully quantify wild-type A-beta 40 and 42 and detect variants within the A-beta 42 sequence. This is a really valuable tool because it enables the identification of familial or autosomal dominant AD, and the automated workflow under which it's run makes it relatively easy to transition right into clinical labs. So now on to a couple of computer science heavy papers, starting with one titled An Efficient Combination Among SMRI, CSF, Cognitive Score, and ApoE4 Biomarkers for Classification of AD and MCI Using Extreme Learning Machine. This is by first author Katari and last author Kwan, published in Computational Intelligence and Neuroscience. So given that AD shows a variable and dynamic progression, this group used a computational approach to clarify the relationship between different AD indicators. To do this, they used an extreme learning machine, which sounds pretty intense, and they included data from the ADNI cohort from structural MRI, including cortical thickness, surface area, and gray matter volume. They also included the classic CSF biomarkers, cognitive scores based on the MMSE, and ApoE4 carrier status from patients with AD, MCI, and healthy controls. Using their computational method, which admittedly I don't really understand, they determined that combining all of these indicators was able to distinguish between groups with accuracies ranging from 83% to 97%, and that the AUC was the highest when all markers were combined in the model. Check out the paper for more details, but again, this illustrates the power of computational methods for improving the diagnosis of AD. Next up is a paper called The Use of Random Forests to Identify Brain Regions on Amyloid and FDG PET Associated with MoCA Score. This is published by first author Zukatinsky and last author Black in Clinical Nuclear Medicine. So the Montreal Cognitive Assessment, or MoCA, is a brief cognitive test used to quickly determine whether an individual is cognitively impaired, and is often used as a preliminary test in the clinical diagnosis of Alzheimer's disease. This study set out to evaluate random forests to identify regions of interest in PET scans that are associated with the MoCA score of the individual. The study included 57 subjects with significant white matter disease, with either evidence of stroke or mild cognitive impairment from early Alzheimer's disease. For the PET scan that assessed amyloid load, the random forest identified regions of interest in the left and right posterior cingulate gyrus, the right anterior cingulate gyrus, and the left and right precuneus that were associated with MoCA scores. In the FDG PET scans to identify metabolically active areas, the random forest method identified regions of interest in the left fusiform gyrus, the left and right precuneus, the left posterior cingulate gyrus, and the left middle orbitofrontal gyrus. The authors also confirmed that amyloid increased and metabolic activity decreased with decreasing MoCA score. Overall, this paper shows that random forests are useful tools to pinpoint clinically relevant regions of interest in PET scans. Finally, I have a couple of smaller studies that involve autopsy data. So the first one is titled Prevalence and Clinical Phenotype of Quadruple Misfolded Proteins in Older Adults. This was published in JAMA Neurology by first author Karanth and last author Abner. So this paper looked at the quadruple misfolded protein phenotype, which is when tau neurofibrillary tangles, A-beta accumulation, alpha-synuclein, and TDP43 are all present in the same brain, a condition that's actually relatively common in aging. The study aimed to describe this phenotype, including trajectories in global cognition, from an autopsy cohort derived from the University of Kentucky Alzheimer's Disease Center Brain Bank. This cohort consisted of 375 participants, of which 76 had the quadruple phenotype. The study found that dementia frequency was highest among those with the quadruple phenotype, and that those in this group also had the lowest MMSE scores, indicating poor cognitive performance. After adjusting for age at death and sex, the ApoE4 allele was associated with higher odds of having the quadruple phenotype. Overall, this study reinforces the fact that AD is often complicated by other coexisting pathologies in the brain, and more work is needed to understand the interplay between them. For the last paper in this episode, we have comparison of amyloid in cerebrospinal fluid, brain imaging, and autopsy in a case of progressive supranuclear palsy. This is by first author and last author Kim, published in Alzheimer's Disease and Associated Disorders. So this study wanted to address the discrepancy that can sometimes be found between amyloid PET imaging and CSF Aβ42 levels. They used a case of progressive supranuclear palsy in which biomarker acquisition and postmortem pathologic examination were conducted very close together, and they found that their case was positive for CSF Aβ42, but negative in amyloid PET, even though Aβ pathology was confirmed in the cerebral cortex through pathological examination. This case study indicates that CSF biomarkers may be more sensitive than currently available imaging techniques for assessing Aβ pathology. And that caps off this episode. As always, thank you so much for tuning in, and I hope you gained some valuable insight into this field of research. Definitely stay tuned for more overviews of CSF biomarkers in the coming months.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.